So welcome to this SPM talk on the general linear model and classical inference. So let's start with an overview of the talk. So first we'll look at SPM for MEG and EEG data and how they're typically analyzed using the GLM, just an overview. We'll then move on to an ERP example to see how we can use a statistical test on ERP data. We'll then define the GLM, especially looking at the design matrix and how it embodies the experimental factors and confounds. And then look at parameter estimation, so how to estimate the parameters of the model. And finally, we'll look at inference, where we use the estimated parameters, the beta weights, to infer effects in MEG-EEG data using contrasts and TNF tests. And we'll also look at the effects of correlated regressors, so the situation where we have collinearity between regressors in the model, and how to deal with that. So first of all, an overview of SPM for MEG and EEG. So usually after acquiring data, we would pre-process the data, as we've seen. So we would filter, we would usually epoch the data into data segments, around the timing of the trials. We would re-reference in the case of EEG data, and we would identify artifacts and remove them. And at this point, we're ready to either average the data in the case of ERPs, or transform the data into time frequency space, for example. So at this point, we can convert these different data features, so be it ERPs or time frequency representations of power, we can convert them into images for analysis in the GLM. So we usually start by defining a design matrix that embodies the experimental factors and confounds. So for example, if we were to compare two conditions, we might use a two-sample t-test or paired t-test, or if we were to compare between groups, say patients and controls, we would use a two-sample t-test. And this allows us to invert the model and estimate the parameters. And using a contrast of these parameter estimates, so C contrast C times beta, we can make inferences about effects, so ERPs or time frequency power, or indeed in 3D source space. And at this point, we would usually also correct for multiple comparisons using random field theory. So let's look at an example of an ERP experiment. So say subjects viewed visual stimuli consisting of images of faces, versus scrambled faces. And we would like to test the hypothesis that there's a difference in the ERP when subjects viewed images of faces compared to scrambled faces. So we would usually take the trial by trial data, so the single trial data, and average them within trials. So average the trials when subjects viewed faces, and separately average the trials when subjects viewed scrambled faces. And we will then take the trial by trial variability and the average and compare the size of the effect, so that's the average, to its standard error derived from the trial by trial variability of the variance. And these two ingredients, so the size of the effect, the average, and the standard error or the variability, these are the two key ingredients of the T statistic, which works like a signal to noise ratio. However, in this example, we have just done that for one channel and one time point. However, usually we are interested in doing this for the entire data set. So we would like to localize effects over various dimensions. So that could either be over time, one-dimensional time, as in the case of ERPs, or this could be 2D time frequency images, where we'd like to localize power or changes in power between conditions or between groups of subjects. Or this could be 3D sensor time images, where we localize over the scalp in EEG, or over the sensor array in MEG, and over time as a third dimension. Or indeed, this could be 3D source images, where we'd like to localize effects in brain space, so in MNI space and voxel space. And this is useful because it allows us to use what is called a mass univariate statistical framework. And this is the framework used in SPM, and what it does is it fits the model, the GLM, to each pixel or voxel in our data images to produce statistical parametric maps, so statistical images. And that's useful because it allows us to localize effects in a broader search space, and it avoids what is called selection bias. So the case where, or the situation where a researcher may look at the data before doing the analysis, and then cherry picking the peaks, say of an ERP, or the peaks in a time frequency representation of power. And in fact, this selection bias, where we would eyeball the data and then cherry pick the effects, has been shown to incur a very large false positive rate. So it's useful to allow the analysis to search for effects and then correct for multiple comparisons, as we'll see in a later talk. Right, so let's move on to the general linear model itself and how it's defined and how to estimate the parameters of the model. So in the ERP experiment, we were interested in comparing faces, responses to faces versus scrambled faces. And the statistical test we used could be formulated as a two-sample t-test. Say in the case of one subject, we would like to compare the two conditions. And this is simply, this can simply be formulated as a regression model or linear model, where we have data Y explained in terms of a combination of parameters times regressors. And in this case, we would have regressors encoding the time points when the subject viewed faces and scrambled faces. So we would have regressors X1 and X2 defining each condition. And we would then have these regressors scaled by a model parameter beta, which in the case of categorical variables, as we have here in a two-sample t-test, these would correspond to the mean, plus some residual error. So in the general case, this is simply a regression model where we have a linear combination of parameters or predicted responses, x times beta, for each parameter p plus error. And we can formulate this in terms of matrix algebra, where again, we simply stack or combine all the regressors into a matrix, and we can estimate the beta parameters at once using linear algebra. So in general, we do this in terms of a data vector y, which is n by 1, so n data points by 1, which is explained in terms of an n by p design matrix with p different regressors encoding p different beta values, model parameters. And in the GLM, these are usually either means, so intercepts, or they can be slopes in the case of continuous variables, such as in regression, or if we include covariates. And then at the end, we have an unexplained error term, which consists of the residuals that are, again, not explained by x times beta, the model's prediction. So this is useful because it allows us to fit this GLM, this model, the same model, to each pixel, 2D pixel or 3D voxel in our data images. And again, these can be either ERPs, one-dimensional ERPs, or they can be time frequency, 2D time frequency, representations of changes in power, or they can be source activities in 3D voxel space, in MNI space. And the design matrix X will then encode all experimental factors and confounds. So again, this can either be at the first level, the single subject level, where we have between conditions within one subject, but usually in MEG and EEG, we would use the GLM at the second level, at the group level, to compare between groups, say, of groups of patients, or and we could include confounds such as age and so on. So let's look at the assumptions behind the GLM and a few conditions that are necessary for the inversion of the GLM, for the estimation of the parameters. So first of all, we assume that the residuals, epsilon, that they're normally distributed and that they are independent and identically distributed. And this is embodied here in terms of the identity matrix scaled by the variance. And then usually it's not the case that X has full rank, which means that X is usually overdetermined. We usually have more data points. So N is larger than P, more data points than model parameters. And in this case, X will be, the design matrix X will be rank deficient. And we will have to use the Moore-Penrose pseudo-inverse. So instead of using the inverse that we would usually use for a square matrix, we use the pseudo-inverse. We can use the pseudo-inverse to estimate, to invert the model and estimate parameters. So for parameter estimation, if the residual error is IID and normally distributed, that's the assumption, we can use ordinary least squares. And this is given in closed form. And it corresponds to minimizing, the solution is given by minimizing the sum of squared error with respect to beta, or with respect to each beta parameter, which again corresponds to setting the gradient of the sum of squared error with respect to each beta parameter to 0. So having estimated the beta parameters, we can form predicted responses. That's X times beta. Those are the responses predicted by the model. And we can then take the difference between the data and the data prediction to form residuals. And these can be used to form the variance estimate. And we note that the parameter estimates, they are normally distributed. So from a geometrical perspective, parameter estimation corresponds to projecting the data the data Y, onto the design space or column space defined by X, for each regressor in X. And that's achieved through a projection. And the projection matrix that projects the data is simply the ordinary least squares estimate beta that we saw in the estimation, pre-multiplied by X. So P times Y corresponds to exactly that, projecting Y onto the model, or the space defined by the model. So this is a linear projection, and the distance in Euclidean space between the data Y and the model prediction X times beta is the residual error. So let's turn to classical inference and look at, in particular, what contrasts we can use in T and F tests to make inferences about effects in EEG and MEG data, and then look at correlated regressors and how to deal with correlations in the design matrix. So a T-contrast is a T-test that is formulated in terms of a contrast of beta parameters, of model parameters. So for example, in the case of two groups of subjects, we could have a contrast of 1 minus 1, and we can then form a T statistic using this contrast estimate. So C transpose times beta hat is known as the contrast estimate. And this is divided by, here, the square root of the variance times a term that we'll look at in a moment. But this T statistic is then used to evaluate the probability that the effect T is equal to or greater than the critical T, the observed T that we estimated under the null hypothesis. And if this probability is small enough or below some alpha value, alpha threshold, say 0.05, we could declare the effect significant. So this is classical inference using the t-test. So if the probability that t is equal to or greater than some observed critical t-value, under the null, if that probability is small enough, small or surprising, we could declare it significant. So let's take a closer look at the t statistic. So we saw that the contrast estimate, so C transpose beta, that's that's the size of the effect, and the denominator of the t statistic is given by the square root of the variance of the effect times a term that scales this standard error. And this term, C transpose times the inverse of the moment matrix, X transpose X times C, is known as the design efficiency, or in fact the inverse of the design efficiency. And it measures how well the design, so X, the experimental design, the design matrix X, how efficient this is for a particular contrast. And at the same time, it scales the standard error that is used in the t statistic. And we also note that the contrast estimate itself is normally distributed. So an example of a t-test is the two-sample t-test. So this would be a between groups design where we would have say, patients versus controls. This could be patients, say, with schizophrenia versus healthy controls. And we could then use a contrast vector, 1 minus 1, to compare the effect between groups. So to test, for example, whether the effect in patients is larger than the effect in controls. And in SPM, the t-tests are always one-sided. They're always one-sided hypothesis tests, meaning that the contrast estimate always tests under the null that, or the null hypothesis is that the contrast estimate is equal to or smaller than 0 because it's one-sided, where the alternative is that the contrast estimate is greater than 0. Another example could be within the same group of healthy subjects, we could have repeated measurements, say 2 different conditions, and we would like to compare those 2 conditions at the group level. That could be scrambled, that could be faces versus scrambled faces. And we would then include subject-specific offsets in the design matrix to estimate the between subjects variance for the repeated measures design. And then we would have a contrast, a linear contrast of 1 minus 1 to compare between conditions and, for example, test whether the ERP to viewing faces is greater than the ERP when subjects viewed scrambled faces. So again, this is a one-sided hypothesis test where the contrast estimate, C transpose beta, is under the null is equal to or smaller than 0, whereas the alternative hypothesis would be that the contrast estimate is greater than 0. Right, so having covered t-tests, we now turn to what are known as f-tests, and these can be thought of as testing a term that we can call extra sum of squares. So in the f-test, we usually test a ratio of explained variance to unexplained variance, where the residual sum of squares, RSS, as we'll see in a moment, is given by the sum of squared errors of a full model, say a model that includes the effect that we'd like to test for, in relation to a reduced model. So this is known as the extra sum of squares principle. So we test for the additional variance explained by a full model in relation to a reduced model. And so formally in the GLM, the way this is done is in terms of a reduced model. So if our full model is X encoding the effects we would like to test for, so this could be 2 groups of subjects, we could formulate a reduced model which is given by the design matrix X times a contrast orthogonal to our main contrast. And this orthogonal contrast is given here in terms of the identity matrix minus C times the inverse of C of the contrast. And we can then form a projection matrix and the residual forming matrices that are then used in the F statistic to compare the variance explained by the design over or divided by the variance not explained, or the residual variance not explained by the design. So the F statistic can be viewed as exactly this. So we have the sum of squares, or the variance, explained by the full model, divided by the variance explained by the reduced model. So if our full model explains more variance than the reduced model, we would have a large F statistic distributed with 2 degrees of freedom, nu1 and nu2, given here by the design degrees of freedom, so the degrees of freedom of the full model, and the error degrees of freedom, so the degrees of freedom of the error estimate or the residuals. So an F contrast can be formed, so we can use a contrast matrix. And this is now different from the contrast vector used for the t-test, because we now have not just one vector of contrast weights, we have a matrix of contrast weights that we can use to test effects. And again, like the t-test, we can evaluate the probability, the p-value, which is the probability that F under the null hypothesis is equal to or greater than our critical observed F, so the one we estimated using our data, our model, and a particular contrast. So F-tests and unidimensional contrasts. So we can also have just a contrast vector where we test the difference between, say, 2 conditions. Again, this could be patients versus controls. However, this hypothesis test is now 2-sided. So the F-test for the same design corresponds to a 2-sided hypothesis test, where the t-test of the same design would correspond to a 1-sided hypothesis test. So in other words, the F value for the same statistical design corresponds to t squared. So it's a 2-sided hypothesis test. So under the null, the contrast estimate would be equal to 0, whereas under the alternative hypothesis, the contrast estimate would be different from 0, so either positive or negative. And this means that these unidimensional tests of parameters, they correspond to, so testing any direction, so either positive or negative. So testing whether beta 1 is different from beta 2, say a group of patients with schizophrenia is different from a group of patients, is different from healthy controls, corresponds to testing the opposite, so whether controls are different from patients. So there's no direction in the test. We would have to use a t-test if we wanted to test for a particular direction of the effect. We can also use what are called multidimensional contrasts. And again, for the same design here, we could have a contrast matrix testing for so any effect of the design, and that will be done here in terms of an identity matrix. So with a 1 over the first group and a 1 over the second group, with ones on the diagonal. And again, we would test the same null, that the contrast estimate is equal to 0, where the alternative would be that it's different from 0. And if we look at more closely at multidimensional tests of parameters, we see that, that this corresponds to testing the alternative hypothesis that there exists at least one beta k, so at least one beta of the beta parameters estimated by the model that is different from 0, so either positive or negative. So it's a test of at least one. So more generally, we could illustrate this with a one-way ANOVA, where we could have 4 different groups. So we could have, imagine we had a group of patients with schizophrenia. We could have a group of patients with bipolar disorder with psychosis, and another group with bipolar disorder without psychosis, and finally a group of healthy controls. And using a contrast matrix, we could then test for any effect of the design. So we could test for any effect in any of the 4 groups. And this would be done using this contrast matrix with ones on the diagonal. And the multidimensional hypothesis test would then look like this. So the null hypothesis would be that the 4 different beta parameters are all equal to 0. There's no effect of any of them. Whereas the alternative, again, would be that there exists at least one beta parameter, so at least one effect in one of the groups, which is different from 0, so either positive or negative. So the effect being either positive or negative. So this is called any effect, or an any effect F contrast. But we could also test for differences between groups. And this could also be done in terms of a contrast matrix, as the one we see here, where again, we have 4 different groups, and we could then test for any difference in terms of this matrix, where each row in the matrix corresponds to a difference. And the multidimensional hypothesis test is again that there exists at least one contrast, so at least one row of this design matrix times beta hat, at least one contrast estimate of all the contrasts, of all the rows, that is different from 0. So that's the alternative hypothesis. So let's take a summary of the GLM. So we've seen that we can use the GLM to formulate both t-tests and f-tests. And in fact, the GLM can be used to formulate all special cases, including multiple regression, analysis of variance, we've also seen. And indeed, if we added covariates to ANOVA, we would have an ANCOVA. So all linear tests can be modeled in the GLM with the right design matrix. So let's look at the example of orthogonal regressors. So usually we would have designs that have orthogonal regressors, meaning that the correlation between x1 and x2 for this design, which is a 2-sample t-test, the regressors are orthogonal, so that there's no shared variance between the parameter estimates estimated by this model. However, we could imagine having a regression model with various predictors, with various regressors, and we could imagine that these regressors, so x1 and x2, are correlated. So this could be a regression model where we would have the mean as x0, the constant, and we could then have x1 and x2 representing, say, positive and negative symptoms in a group of patients with schizophrenia. And estimating this model will result in beta parameters due to x1 and x2 that are not unique. So they share variance because x1 and x2 are correlated. So when testing for x1 using a contrast with a 1 over x1 and zeros elsewhere, we would in fact only explain the part of the variance, the part of the variability that is not explained by X2. So that would be not the full sort of space or variance explained by X1. It will be the part that is not explained by X2. And vice versa, when testing for the effect of X2, that could be negative symptoms. If positive and negative symptoms scores are correlated in this group, we would only be testing for the effect of X2 that is not shared with X1. So we could, for example, orthogonalize the regressors. So we could orthogonalize X1 and X2, and this would result in X1 explaining well its full variance, and X2 then only explaining the part of the variance that was not explained by X1. So in general, when we orthogonalize regressors, what we do is we remove the part of say X1 that explains X2. So in a standard regression model, for example, we would usually mean center the continuous regressor, and this in fact corresponds to removing the part of the intercept of the constant X1, and this happens to be the mean. So in general, orthogonalization removes the part of one regressor, explained by another regressor. So they become effectively orthogonal. So if we orthogonalize X2 with respect to X1 in a regression model, what happens when we estimate the parameters is that we get unique parameter estimates. However, the parameter estimate that changes is not X2, or the parameter estimate due to X2, so it's not beta 2. In fact, it is the parameter estimate of beta 1 that changes. And this is because we orthogonalized, in this case, X2 with respect to X1. So this means that X1 will now explain its full variance. And this is why the beta parameter for X1 changes. And finally, we could have the case where we still have correlated regressors. So X1 and X2 are still correlated. So positive and negative symptoms are correlated in the model, they're both included. But we could actually still test the full subspace of a contrast. We call that XC. So testing that full subspace would require us to formulate a contrast C as the moment matrix, so X transpose X times the contrast. So this will in fact test for the entire, for the full subspace of the contrast. Right, so let's summarize what we've been through. So we saw that we could, after pre-processing, we could convert our data features to data images. And we could then enter these images of different dimensions into the GLM, estimate the parameters using a design matrix, and then use a contrast, either a T contrast or an F contrast, to make inferences about effects in time in the case of ERPs, or effects in time frequency space in the case of time frequency data, or indeed effects in 3D voxel space in the case of source reconstructed data. And at this point, we would usually correct for multiple comparisons. So this concludes the GLM talk, and thanks to everyone, to you, and the SPM Methods Group and collaborators.